Hey there, we are back for part two. Video worked fine, so hopefully they'll keep working fine. It's just my computer messes up every time I try to get the slideshow on this screen. It keeps going to my other monitor over here. So I have to go through this whole thing about swapping slides and swapping screens. So proteins. So part two here, proteins can be used for tons of different things, like literally tons of stuff. So it could be structure, okay? So it could be things like muscle fibers, could be things like hair for structure. Your skin is filled with a collagen protein, which is what makes your finger stretchy. Why you can take the back of your skin and do that and pinch it up because the collagen is in there. Hooves and nails are made of proteins. Okay. Spider silk made out of protein. So this is a table from your book. It actually might be from the old book. Either way, I don't need you to memorize it. It just kind of shows you all these different things over here on the left that proteins are involved in. They're involved in a lot of your genetics and how things get, genes get expressed. Motor proteins, for moving cellular components, defense proteins, metabolic enzymes, self-signaling, structural proteins, and then transporters. Like almost everything that goes on in your body is somehow involved with a protein. Okay. So proteins are made of chons. Okay. So far, carbs were made of CHO, lipids were made of CHO with a little o, and proteins are made of chons. Okay. So if you notice from your pattern matching, my pen never really works that well when I make these videos at school, but I'm going to try a little bit here. If you notice with your pattern matching, you'll see an NCC backbone. Okay, with all of your amino acids, you're going to see NCC, NCC, okay, NCC, NCC. So you'll always see that backbone, and that's where a bunch of your amino acids should have been grouped in your pattern matching. And then guess how they go together. Look at this red area right here. You have an H from this one and an OH from this one. So how do you think they're going to join? Same thing as everything else, right? Dehydration synthesis. You make the extra water molecule, and you're able to put the two amino acids together. Okay, and then amino acids, or I'm sorry, proteins, have a special bond type called a peptide. I was really hoping that line would draw, but it's just not drawing the line for me. But hopefully you see where my pointer is at down here, bipeptide bond. Anytime you see peptide bond, that refers to protein. Okay, so the bond in between each of the amino acids is always called a peptide bond. And in fact, proteins are often called polypeptides because it takes a lot of peptide bonds to make a protein. Okay, so polypeptide's kind of another name for a protein. Got to get rid of the pen before it lets me switch. The only thing I don't like about this. So the monomer for proteins are amino acids, okay? And the bonds are called peptides. So same thing we just kind of talked about. Proteins are made of amino acids. So here's the thing is you have this backbone, right? Here's your NCC backbone, and you have some other things hanging off. This end down here, those three are your amine group. And this end over here, COOH, that is your carboxyl group, okay? So that is really, you're going to see things called an N terminus and a C terminus. So whenever you string amino acids together, there have to be like, the ones on the end have to not be attached, right? On the opposite side, the free side. So at one end of the protein, there will always be a free amine, and that'll be your N terminus. And at the other end of the protein, there'll always be a free carboxyl group hanging off, which is your C terminus, okay? And we do this just for communication purposes and being able to talk about the protein orientation and stuff like that. More importantly is this R group, okay? So you see the R group hanging off, okay? So the R group is interchangeable, okay? The R is just a placeholder for other functional groups and things that will attach there and make every amino acid then different, okay? So There are only 20 amino acids in the world, but you can make a lot of different combinations with 20 things, especially when you can vary the total amount of amino acids that you'll use. Some proteins are as small as 10 amino acids, others take thousands. And so when you have 20 things and you have the ability to vary how long those are, you can make an infinite almost number of combinations. Okay. So if you look at the slideshow here, you can put an H on there, that's glycine. If I switch out the H and I put that thing on there, that's leucine. If I switch it out again and go with that, that's phenylalanine. So the rest of it stays the same. The whole stem, the whole backbone stays the same, but that R group is interchangeable, and that's what makes all the different amino acids. Okay, so again, we join them, we pull out the water, and we do dehydration synthesis. Okay, so for every peptide bond we do, we pull out of water and we make a peptide bond, okay, between one amino acid and the other. So here is a small amino acid, and the question is, how many peptide bonds do you see here? So go ahead and pause the video and try to come up with how many peptide bonds you see. All right, hopefully you pause the video and I will come through here, and NCC, that's a peptide, NCC, that's a peptide, so it's 2, 3, 4, 5. Okay, there are 5 peptide bonds for these 6 amino acids that are strung together. Okay, so hopefully that makes sense to you. And here's what I meant by the N terminus and the C terminus. You can see on the left-hand side, the free amine group is hanging off the end there, and on the right-hand side, the free carboxyl group. So C terminus, N terminus, just the free, whatever's hanging off the ends of those amino acids, and those stand for the amine and the carboxyl groups. And it's really just, again, for communication purposes. So if there's a protein that's 600 amino acids long and you're trying to communicate with a scientist about a specific portion of it, you can say, you know, the amino acid that's 40 in from the C terminus, okay? And they will be able to orient themselves a little bit better that way. So a protein is made of generally, you know, 100 to 300 amino acids. You can definitely get much smaller and you can definitely get bigger, but a lot of them are in this range, okay? And again, many of them are called polypeptides. And if you can stop and really squint a little bit, you can see all of the writing on these little purple circles over here are the abbreviations of all the amino acids, okay? So, sorry, the way your proteins are developed is based on the order of these amino acids. So getting the amino acids in a particular order is key to proteins working. If they get out of order, then that protein's not going to do its job, and then you'll have a problem. You'll have some sort of phenotype, right? You could have a disease, you could have a disorder. It could just be something that's minor and doesn't work very well. We all have blemishes like that, and we don't even know it most of the time because we can get along with a partial mistake. But sometimes you have really bad problems when you get even one thing wrong. Sometimes you can have a pretty bad genetic disorder. So here's an example of one of these. So sickle cell anemia. So this shows the sequence of the amino acids, and you can look up top, number 6 there, glutamine, up top on the normal has been swapped out because it has the wrong amino acid in the sickle cell one, and that comes from the DNA code having some sort of mutation and putting the wrong amino acid there. And so it got valine instead of glutamine, and now instead of your red blood cells being nice and plump and round so they can carry a bunch of oxygen for you, they look like that bottom picture right there, and they can't carry a lot of oxygen. So people that have sickle cell would not want to travel to Colorado. Okay, they would not want to play really. They would not want to be maybe a runner. Aerobic sports could slow them down a little bit. Some cases are more severe than others. There's actually a guy in the NFL that I know has it, and the team will not let him play when they play in Denver. He has to sit out because it could endanger him. So that's kind of an example of when having the wrong amino acid, just one wrong amino acid, can really give you a problem. Here's another one, not necessarily oops, not necessarily a one-off, but having amino acids out of order can definitely cause problems. And so this is why we are trying to figure out the order of all of these proteins. So it started actually with the Human Genome Project, which I got to work on in the years like 2000 to 2000 and oh, I was probably there in actually 98. From 1998 to 2003, I worked on the Human Genome Project at WashU, and we figured out the order of all of the ACGs and Ts in DNA. And that order then dictates the order of the proteins, the order of the amino acids. And so science has been trying to put all these puzzle pieces together ever since to try to figure out, what's causing certain disorders. And so once we figured out the human genome order back in the early 2000s, then a lot of discoveries have been made based upon that. So it's a pretty cool project to be a part of. So Hutchinson-Gilford progeria syndrome, it's advanced aging. These kids are like 9, but they look like grandpas, right? And so just kind of a different syndrome. But again, faulty proteins can give you these disorders. So proteomics, by the way, is the study of all of the proteins in your body and how they kind of work as a whole. And so you might have heard of genomics a little more than proteomics, but proteomics is a big field. So there are different levels of structure, kind of back to a similar picture here. And the order of the amino acids themselves, the basic order of the amino acids, that's the primary structure of the protein, okay? So if you look up here, grab my pen again, you have lysine and glutamine and threonine and alanine, another alanine, lysine, phenolphthalein. So this order, okay, the snaky order of our protein right here, that's the primary structure, okay? How would a cell know the correct order to use? Well, I kind of told you about that a second ago. It's based on the order of the bases, the ACGs and Ts, that's in DNA, okay? That's really, the DNA is the instructions. And so that's how we're going to know this. The secondary structure then, if I bring this up, the secondary structure, once you have this primary structure, well, amino acids are charged. Many of them are positive and negative. And so the positive and negatives will actually attract to one another, and then the negatives and the negatives and the positives and the positive will repel each other. And so certain parts of the protein want to wrap and fold together, and other parts want to stay away from each other. And so you end up with these secondary structures, and you'll see things in here. You'll see, wish I could draw a line, but I don't think it's going to let me. But I'll put dots right in here. That's a helix, right? You can see the spiral. So you have those, and you'll have another one called a pleated sheet, which I'll show you in just a second. But these are secondary structures that are based on how the amino acids attract or repel one another. There's an example of the helices, which I just showed you, the helix, which I just showed you on the other slide. But the top one up here is the helix, and then the bottom one is called a pleated sheet or a beta sheet. And so you get these interactions with them. And so those are your secondary structures. The tertiary structure, tertiary being another word for like third, okay, is the overall structure, right? The shape of the entire protein. And they all are different, okay? All these proteins are different. But basically, the whole thing, the whole shape of the protein is the tertiary structure. And this is important because proteins interact like jigsaw puzzle pieces, okay? Generally, proteins interact with receptors and other things, and they have to like fit together like perfectly. Like if I put my hand like this, a protein like this wouldn't fit in to my hand, right? But a protein like this would fit in. So the puzzle pieces have to fit exactly together. So the overall shape is very important, right? Structure and function. And then finally, quaternary. This is when you have multiple proteins, okay? So you kind of, there's a protein up, oh gosh. So there's a protein here, there's a protein here, protein here, protein here. And when you put them all together, then you see how all 4 of them have come together. That is a quaternary structure when there's like interaction between multiple proteins, okay? And this has to happen. Some of them won't work by themselves. They have to work as a team, okay? So again, just a slide as a review for the structure. Here's your primary, which is just the list of the amino acids. Here's your secondary, your helices and your beta sheets. Here's your tertiary, which is the overall structure, okay? And then over here is the quaternary, where you have multiple proteins interacting together, okay? So proteins can also be enzymes, okay? So not all proteins are enzymes, but all enzymes are proteins, okay? Hopefully that makes sense. Enzymes are there. You probably know this about enzymes already. They're there to speed up reactions, right? They're the catalysts, okay? They're used in just about every reaction that we have. So as a reminder, grab a pen. As a reminder, kind of just go through some terminology here. You have your enzyme. That's the big purple thing here, okay? Big purple guy is our enzyme. We have active site where you can see this groove in here, okay? That's our active site. That means that's where the protein's going to work on a substrate. So our substrate is sucrose, and sucrose, as you know, is a disaccharide, okay? So our enzyme is called sucrase. Again, no, oh, there it goes. Let me make a line. And if you look over here, our substrate is sucrose. So enzymes are always named after what they work on. So sucrase will break down sucrose. And remember, the oses are carbohydrates and the aces are enzymes, okay? If you ever see ASE, that's always an enzyme. So down here, our sucrose is going to bind right here in the active site, okay? And then the enzyme is going to do its job, and it's going to break that bond, okay? And we're going to have to use some water, right? Some hydrolysis here to break things apart. And then up here, you can see now we have glucose and fructose, which are the monosaccharides that build the disaccharide, okay? And then potentially another enzyme will then put them together, and then it could repeat. Just depends on what's going on. But it ran into sucrose, so it wants to break that down into energy, okay? Into smaller bits that it can use. So hopefully that makes sense for you. And it's getting stuck. This happened yesterday. Sorry, it's lagging and it won't let me get rid of the pen. So hang on real quick. I can't get to the pause or anything. Aha. Now I think I can move on. So certainly a direct relationship, big time relationship between structure and function with proteins, right? It's all about its conformation, okay, and its function. So a lot of times we have proteins that change and they get a conformational change based on the environment, okay? And so a protein could become denatured. When a protein becomes denatured, it generally doesn't work, okay? So what could cause a protein to denature? Okay, so think about some environmental factors that proteins would have to deal with in order to work. So pause the video for a minute. What might affect the protein? A protein's ability to do its job. What sorts of things will it run into that would maybe change its conformation? Wasn't sure if there was another slide or not. Several things, actually. Temperature could affect it, right? So think about eggs, right? If you go to make eggs, eggs have protein. You guys know that, right? You eat eggs for protein, okay? So you scramble an egg, okay? Can you unscramble an egg? Can you make it go backwards? No, because you denatured all of the protein when you heated it up. So the proteins became denatured, and now you can't put the genie back in the bottle, right? It won't go backwards. And so that's one way. So temperature, pH, salinity, different ions, different positive and negative molecules will make those amino acids then pull in different directions. And if it changes shape, it might not work at all. It might work partially. It might have no notice. Like, it just depends on what part changes. But that can be a big problem for proteins. So I'm going to pause it here or stop it here, and then I will be right back with the nucleic acid portion.